Hello Cypress Grove, my name is Mr. Nelson. I'm going to be your resident scientist this next year in Miss Beckhelm's science class. But first I want you guys to learn a little bit about me. This is the building I work in. It's the Animal Science Center. It's called Clayburgh Center. It's named after this man on this statue here, Robert Clayburgh. He was the chief executive officer of the King Ranch for many, many years, and to honor him and all the research that he did in animal science, they named this building here at Texas A&M after him. So if you guys want to follow me inside, I'll show you what I do.
Here we are now in the atrium of the Clayburgh building where I work. You can see there's a lot of trophies and awards behind me, and they're scattered throughout this building, and they represent different things that judging teams have won around here. The bronze bull that you saw or see a picture of is actually the traveling trophy for the national champion judging team, livestock judging, and A&M's won that the last 3 years. I was also a part of the team in 2001 that judged here. Although we didn't bring the bull home that year, we ended up fourth. However, it's a really long-standing tradition here at Texas A&M with our judging teams within our animal science department. What really got me into science, I guess, has to do with livestock, and that I grew up in a real rural type community in a town without a population sign. And pretty much on the whole way to school it was farms and ranches, and that's all there was. And it's kind of always been an interest of mine. And then as I got older, I got more into different types of reproductive techniques and got really interested in the science behind reproduction, and that's kind of where I've ended up where I am today. And I guess the whole goal for me is that I'd like to maybe end up teaching at a smaller type teaching institution, reproductive physiology type classes, or my other alternative that I probably would enjoy even more than that would be to have or work for an embryo transfer type company that did assisted reproductive techniques and different types of things like that, just because I'd always get to be outside and working with different producers within the business. Well, now we're in a typical animal science classroom here in the Clayburgh building. You can tell this is a lot bigger and a lot bigger scale than what you guys are used to sitting in. This classroom right here actually holds close to 160 students. I sat in the same classroom several years ago as an undergrad, and now as a graduate student, I sit in here in TA classes for Dr. Forrest, and every now and then, in case of his absence, can lecture in this classroom. But it's a lot different. You can see it's a lot bigger room. There's a lot of students packed in here, and all of these chalkboards, there's actually 4 of them back here, will just keep raising and lowering. So whoever's up there writing can just start writing and send that chalkboard up and keep on writing on something else. So he's not real limited on space. It's a lot different experience when you're in a classroom this size. You really have to pay attention. There's not a lot of personal attention directly at you, and that's how it's a lot different than where you guys are at right now. You just kind of have to take care of your own business because nobody's really looking over your shoulder anymore. Guys, I think we're in luck. I think I just caught Cindy in here, and I think she's running a gel, and this should be pretty neat for you guys to see, so come on in. Hey Cindy, what's going on today?
Hey John, I'm running a gel.
Okay, what kind of gel are you running?
I'm running a DNA gel, and it's on a certain agarose concentration.
And what kind of results will you be looking for later?
Well, I'm looking for 100 base pair fragment.
So when you say base pair, what are you talking about?
Well, based on the marker that we can show you in just a minute, you'll see where that 100 base pair hits running down the lane of that gel. And if the cloning project was successful, then we'll see 100 base pair fragment.
So we're looking essentially just for size, right?
That's correct.
Okay.
Alright, I'm getting ready to load these wells. I've got 5 samples, and I'm on number 4. I'm loading 10 microliters, and they've got some DNA dye in there so that we'll be able to see the samples under the ultraviolet light. And that wasn't a very good loading job. Since that one too questionable about that one, we're going to reload number 5 again. Very good. Okay. Put the lid on it. Black to black. Red to red. all the current will run towards the positive, which is red. Hook it up to the power pack, black to black, red to red. And that way all of the DNA will be running towards the red current circuit.
Well, Cindy, looks like we're all done. I guess now it's time to see if you got the right bands.
Yes, we're going to put it on the UV box.
Okay. How long did this take?
It took about an hour to run at 125 volts.
Okay.
Now we've got it on the light box under UV light, and this shield is protective for your eyes so you don't burn them. And what we're looking for is 100 base pair, which is down here by this big blob of RNA, which is really what we don't want. And the 100 base pair lines right in there. They're real hard to see because it's so small, but we have this PGM marker that tells us where the 100 base pair should be. And I can see that we've got two good clones out of this vector, and hopefully that'll be good enough to stop at this point and sequence the results and make sure that we can identify the product.
Well, guys, now we're in here with Katie, and she's a PhD student in a reproductive biology lab, and actually they focus on the uterus. And she's going to show you some really neat images of some things that she's been working with recently.
That's right, John. My lab does work on the uterus, and we look at a lot of different things, one of which is RNA. And RNA is part of the message for a cell. One way that we do that is to actually use radioactive material. Now, it's very safe, very low levels, but it's great because it really helps us to show where the RNA is working. So what's on the screen here are actually two images of the exact same thing. This is a really thin piece of uterine tissue that we took a picture of, and you can see the dark and white specks. Well, because we've used radioactivity, that will actually glow when you turn out the light. And this is a picture showing all of the areas where RNA is being processed. So from this one technique, we can get a picture of the tissue and an idea of where the RNA is working.
So what are all these little bright circles out there, Katie?
Well, the bright circles actually, John, that's where you can see a lot of signal, and that's because these are what are called uterine glands, and the glands are very important for producing a lot of things to help the uterus grow and develop. And so they're very busy, which is why you can see so much RNA packed into those points, as well as this layer of cells along the outside.
Why do we care about RNA in terms of the uterus and physiology, or what are we looking at?
Well, RNA is really critical for any cell to do its job. As I'm sure everybody's heard of DNA at some point, well, RNA is the next step in the process. It's part of the blueprint, if you will. So this is part of that message.
Sounds pretty interesting, Katie. Well, now Katie's got a really interesting looking picture that's quite a bit different from the last one. So Katie, what exactly are we looking at now?
Well, John, last time we were looking at RNA, so that was just a very, very small picture. Now we've actually got the same tissue, but we're looking at all of the cells. And as you can see, there's a lot of different colors involved here. And that's because the stain that we use for this, each cell type will stain a different color. So what you see here, these big red stretches, those are actually muscle fibers. So by the stain, we can tell that there's muscle. And if you can look here down on the base, that's actually a blood vessel. So the cells that surround the blood vessel are stained a little differently because they have a different job.
So are there any other types of cells that you can show us on this slide? Or?
Well, sure. Because what you get to see on the computer, I can also see through the microscope. And if I just use the stage control here to my right, we can move and look at different parts of the tissue. So if I go just a little bit over, get that back into focus for you, you can see how the tissue looks very, very different. Well, it's the same piece of tissue, but now we're not looking at muscle anymore. We're actually looking at those glands, which when we were looking for RNA, those were the bright spots. Well, here you can see each different cell is colored in that dark red purple, with the area, the open area in between, being more blue in color. So this stain, this trichrome, because it uses 3 main colors, gives us a better picture of what kind of tissue we're looking at. So it's another tool to evaluate a cell and a tissue in the body.
Well, there you have it, guys. Well, I'm going to show you guys now what I do as far as my research or parts of it. I don't actually work in one set lab. I work what we call on farm. I work under a professor who owns an embryo transfer company. He's an adjunct professor, and so many times I'm not actually in this building at all. But I set up a camera and microscope to show you guys kind of what I do. First of all, this is a straw, and these are usually stored in liquid nitrogen. And in this straw, you'll see there's 2 main compartments and then a third one right in the middle. This is where the embryo is actually kept. And so it's frozen like this, and then what I've done is actually thawed an embryo to show you guys under a microscope what I look at. Now these are actually cow embryos, and this is a common practice. So what I do now is we'll search in this dish and try to find the embryo. Okay guys, now I've actually found the embryo, and I'm going to zoom in on this embryo and show you guys a closer picture. And you can see exactly what I'm seeing right here on the computer monitor as I zoom in on this embryo. Now it's in the very center of your screen coming up. And this is actually an embryo that's not actually perfect anymore. It's kind of degenerated, probably because I've had it out for a few seconds while I was talking and didn't immediately get it into the correct transfer medias. But that is an embryo for you guys, and it's a bovine embryo that was taken out. It could have been taken out 10 or 15 years ago, but it's been preserved all this time by being frozen in liquid nitrogen. Well guys, now we're here in my office. Y'all saw the front of the building, and this is the inside where I spend quite a bit of time when I am here on campus. You can see my desk pretty cluttered. That's kind of how it stays most of the time when I'm here. Most of these things that are covering it are journal articles or different presentations other people have done. And right now you can see I'm working on a presentation for one of the professors on my committee that he'll actually present in Australia here in about 2 weeks. One of the interesting things about being a grad student, I guess, is that there's a lot of different people that you meet that come from a lot of different places. Here there's 8 of us in our office, and I think 4 of the people are international students. There's 2 from Colombia, 1 from China, and 1's from South Korea that I share this office with. So it's not just Americans or white Americans from rural backgrounds that are in animal science. It's people from all different types of backgrounds that work up here. Our section's actually reproductive physiology. And what we do is focus on basically increasing reproductive efficiency, whether it be in farm animals or humans. All the biology is the same. So a lot of these animal models are used for later experiments that are done in humans, but we have to start experiments, usually begin at the rats, work our way up through rabbits, then into sheep, and then into humans and things like that. And we're kind of at that level where we do the different things in the livestock species. And that's what most of the people that you'll see later on in this video actually work in the sheep. The other thing that I do a lot of is I travel. Up here on my calendar you can see all the days that have something red in them are days basically that I'm out of this office. And I work mostly at an embryo transfer clinic for a professor that's on my committee, and that's where my research is done at. So you can see usually 2 or 3 days a week I'm not here at all. And then 1 day a week I'll actually be with you guys in middle school, so that only leaves about 2 days that I'm actually here. So I'm never here just stuck in the same place being too bored. There's always something changing. And when I do go out, usually we go out on different people's ranches, and that's where we do the embryo work on cattle. So I could go 2 or 3 hours north of here, 2 or 3 hours south. It just depends on who's got a job for us to do that day. What I mainly do is I focus on embryo transfer. That's my line of research. And what that involves is actually taking embryos by forcing a cow to have more embryos than she would normally have, because usually you see a cow that has one calf. What we do is try to make her have more than one calf because she's superior either genetically or phenotypically, and we want to have as many babies from her as we can. So what we do is increase the number of embryos she has and then take those embryos and place them into other cows that we call recipients. So that one cow, instead of having one a year, may have anywhere from 5 or 6 up to maybe 30 calves in a year, because we put our embryos into other cows. And my focus is increasing the efficiency in the number of calves we get. So all those embryos we put in aren't always pregnancies. And that's what I work on, is to increase the number of pregnancies we get and increase that percentage. So that's kind of the basis of my research. Now, when I'm not here at work, there's a lot of other things I do as far as for fun or things I enjoy doing. And one of them actually relates to what I do here, and that's how I pretty much got interested in what I'm doing, is that we run cattle ourselves. My dad and I own quite a few together, or not a whole lot, but enough to keep us busy. And this is one of mine, a Red Angus female that I bought, and she's one that we'll actually flush and transfer embryos out of this next year. So our goal is really to raise kind of show steers that kids show at county fairs, or purebred cattle that people show for us. And that's kind of what we focus on, and we have enjoyed doing together quite a bit. Another thing I enjoy doing, I like going up places like Wyoming and Montana. And this last summer, I got convinced to be in a wild horse race up in Wyoming, and you can see the horse got the best of me a few times, but I like doing things like that. It was pretty neat to go up there, pretty interesting. You know, Wyoming's hardly as populated as it is down here, but it's a real interesting place, real small town, and that's kind of the type of place I'd like to actually end up in one day. But just so you know, in case you're interested, we did get her saddled eventually and get her rode across the finish line. It just took us a little while longer than the rest of the guys. So these are some just different pictures that I took when I was up in Wyoming this summer. I actually got to go up there for about 10 days and camped up in the Bighorn Mountains, and that's a picture down there where it kind of goes down and you start seeing the pine trees is where we threw our camp up and stayed down there for 4 or 5 days, I guess. And I pretty much just caught fish and lived off of that and ate fish. And this is my old roommate before I actually got my master's. I'm on a PhD now, but before I did my undergraduate at Texas A&M and did my master's at New Mexico State. And this was one of my roommates while I lived at New Mexico State, and we've remained really good friends. So I went up and stayed with him, and this is him. This is the creek that we caught all our fish in. We fly fished pretty much every day we were up there. And so these are just different pictures of that creek. And the real neat thing about this place was that he pretty much guaranteed me we wouldn't see a person when we were up there, and we didn't. We were pretty much miles away from where anyone would dare to camp. So it was pretty nice to get out there, not hear any cars or see any planes for that matter, because of the place we were at, and for sure didn't see any people. So it was a really good trip, and I just enjoy being places like that. And here's me deciding or figuring out that at 8,000 or 9,000 foot elevation things don't work the same as they do here at sea level, and Craig thought he was pretty much going to have to carry me back to camp because I wasn't in near as good a shape as I thought I was in. So that's me almost dehydrated, and that's us getting real close to camp and me finding some water finally and making it through the day. So that was kind of my summer vacation. Those are the things I really like to do. I'm into the outdoors a lot. I fish, I hunt. Anytime I can be out of the office, I'm a lot happier than being in.